Thank you for the introduction. Last year, I just introduced the microstructural asymmetry features, and this year I will talk about the heritability and psychopathological and their behavior association. And now we know like brain lateralization is a fundamental principle in life. For example, when you perform a specific task, your brain will prefer to behavior in the left or the right hemisphere. For example, for the language, the left hemisphere is more activated than the right hemisphere. And to wonder how this brain lateralization happens, people are wondering if there is a very specific neural population much better, higher than in the left than the right. So people can study the cortical thickness and the surface area across the cortex. And for the surface area, people find the posterior regions are more left lateralized, and for the posterior areas, they are more right lateralized. And all the asymmetry features can be heritable. To be more specific, people are wondering whether the myelination can be left lateralized or right lateralized. So Glasser studies using the HCP data, studies the group level T1, T2 ratio lateralization. They find the posterior areas still present very left lateralized, and for the posterior regions, they are more right lateralized. But after the transmit field correction, the asymmetry become less, but there still exists. So for the post-mortem data, people also find for the psychoarchitecture in the left hemisphere in the BA area 45 language region, there showed a higher intensity in the left than the right hemisphere. More recently, and Casey Piccolo developed a way to study the global feature of the intensity, and they using the microstructure profile covariance to describe the organization feature across the cortex, and they find the in vivo HCP data and also post-mortem big brain data. They follow the first eigenvector follows a sensory to fugal axis. So far, the structural asymmetry is only considered over the surface. However, whether this asymmetry can vary along the cortical depths is still under studied, and how the microstructure profiles are organized differently between the left and the right hemisphere still also under studied, and whether this kind of microstructure asymmetry is under genetic control. And finally, I will study the depth-specific behavior association. So basically, we focused on the big brain data for the psychoarchitecture asymmetry and HCP data for the in vivo microstructure asymmetry. And we also use MICA-mix data to do the replication. And I will show the big brain and HCP in this presentation. And for the surface reconstruction, we use the 6-layer estimation from previous study and reconstruct 10 equivolumetric surfaces for the big brain. And so in total, there are 60 surfaces for the big brain, and 12 surfaces for the T1, T2, and 40 surfaces for the QT1 MICA-mix data. And for the asymmetry index, we just use left minus right. And based on the HCP trend-based pedigree, we can calculate the heritability of the microstructure asymmetry. And for the global feature asymmetry, we use the gradients approach, is a diffusion map embedding on the microstructure profile covariance. And finally, we use machine learning to study whether the layer-specific asymmetry can be correlated with behavior. So first, I show the intensity asymmetry. Yeah, just consistent with previous studies, posterior areas showed a very right lateralized intensity, and for the anterior part of the brain, there showed like left lateralized asymmetry. And the results are consistent between the big brain and HCP T1, T2 ratio. And for the layer-specific asymmetry, we find that for the big brain, the asymmetry can vary along the cortical depths. For example, for the language, the very lateralized region, the superficial layers there showed very high left lateralized feature. But for the deep layers, they showed a little bit like right lateralized asymmetry. And for the in vivo HCP data, the language networks, they showed same direction for the left lateralized asymmetry, but it also showed a little bit variation along the cortical depths. So to understand this variation, we calculate the coefficient of the variation. It's just an indicator estimating the level of dispersion of the data. And we find for the language regions, it showed quite or high variation in the cortex. And the most peak variation happened in the ventral multimodal network. And for the in vivo data, it showed like as somatomotor network and the default mode network, they showed a very high variation. And then to study the global feature, we use the gradient approach. Yeah, everyone knows it. And we focused on the sensory fugal axis to understand the global feature of asymmetry. And then we find for the language areas, they're more like left structured than, or just like comparing the laminar structure between the left and right. We find language also have a high laminar structure in the left hemisphere. And to understand how this asymmetry distribute along the eigenvector one, the sensory fugal areas are more have a higher laminar differentiation in the structure, and for the fugal areas, they showed a weak laminar structure. And we find in the middle areas, they showed a more higher left structure, but for the two ankles, they showed a right structured feature. And then to calculate the heritability, we find all the asymmetry features can be heritable across the whole cortex, and more specifically, we also calculated the heritability for each different layers. And here, using HCP data, every 3 surface is as a cortical layer, and we find the middle layers heritability is much higher than the lower and upper layers. Then, to understand this asymmetry feature can be correlated with behavior, we study the very lateralized function. First one is psychopathology. And because for the asymmetry features along the cortical layers, they're very similar, so we use some machine learning pipe, the ridge regression and lasso regression to see how these collinear features can be zeroed with the increasing of L1 ratio. And we find for the psychopathological prediction, we find the deep layers can be used to predict the psychopathology in the high lasso ratio. And then for the language, we find, firstly, we find all features can be entered to the model to predict the language. And with increasing of R1 ratio, it shows like there is no trending for the superficial or deep layers. So to summarize, asymmetry varies along the cortical depths with a high peak as anterior temporal gyrus and also somatomotor cortex, and a low peak at the secondary visual cortex. Along the laminar structure, sensory fugal axis language are more left structured, but posterior multimodal areas are more right structured. Microstructure is heritable in general, but more obvious in the middle layers. And in the wide association between microstructure asymmetry and behavior, selected features are more centered on the deep layers for psychopathology, but decentered for language features. That's all. Thanks for the support from the collaborators.
I think we have time for one quick question. Yes. Right.
Lovely presentation. I've got a question. It's not up now. Something like your third last slide, you had a measure where you said that superficial larger than deep, larger than superficial minus deep equal deep minus superficial. what do you mean with these superficial deep? Are you talking about the relative width of the supragranular,
Yeah,
infragranular layer?
is more close to the pure surface.
Yeah.
Yeah, and the deep layers is more to white matter. Yeah.
But what aspects of these parts of the cortex were you comparing when you had this? Were you talking about the thickness or the relative width of the layers, or were you talking about the cellular density, or so what?
Density, yeah. The intensity.
Intensity.
Yes.
Right. So that would be intensity in the right cell packing density. Thank you.
Thank you so much.